right, the next team is the professionals, a team of two. The customer is Professor Densmore, here for you. And their project is Automated DNA Assembly Platform for Bioengineering. So this is the professionals.
Hello, everyone. We are Team 16. Our project is Puppeteer and Automated DNA Assembly Platform for Bioengineering, and our customer is Professor Densmore. So what I'd like to do first is give you a bit of background. And what our project targets is synthetic biology, more specifically, the needs of synthetic biology. What I have here are a couple quotes from two organizations. SynBIRC was the first federally funded synthetic biology research center, and SBC at MIT is a recent organization that was designed to develop and advance the engineering discipline in synthetic biology. And in a nutshell, synthetic biology involves the engineering of genetic organisms or biological systems with targeted behaviors. So here are some examples of applications of synthetic biology. And this ranges from during the early years of when synthetic biology research emerged, up to the current years where there are revolutionary systems being designed with synthetic biology. And this kind of will exemplify why we chose a project that targets synthetic biology. So our first example is a genetic toggle switch, which basically mimics the cellular form of memory. The second example is known as a repressilator, which is a biological oscillator. Our third example, which is a little more complex, is tumor-sensing bacteria, which employs techniques of biosensory applications and biotherapeutic applications, where bacteria targets cells that have components that register it as cancerous. And the last example is antimalarial drug administration, where through a synthetic biology pathway that was discovered through the efforts of some of the big names in synthetic biology, that lab was able to develop a technique to produce large amounts of artemisinic acid, which is required, a precursor for the current pathway for solving or for dealing with malaria. And additionally, synthetic biology also has significant applications in bioremediation and biofuels. So what exactly is the basic procedure in synthetic biology? We start with DNA assembly. And DNA assembly involves building genetic constructs. It allows you to build larger genetic constructs through the composition of smaller genetic sequences, which allows for engineered mechanism behavior. And DNA assembly is a manual procedure that involves planning, optimization, resource computation, and time. And the three specific types, I've mentioned three of them, and thus two of them. And the last type is the actual execution of these planned procedures. So what I'm showing you here is data collected by the Synthetic Biology Project that shows global synthetic biology research conducted in 2009 and 2013. And it's just a comparison. What I want to highlight is how much synthetic biology research has grown. Specifically, you can look at the university category. You can see that there are over 200 university organizations that perform synthetic biology research. And this number is growing. And the majority of this research actually involves manual DNA assembly. This is performed by hand, and it's therefore not scalable, and limits their capability to build larger genetic constructs.
Right, so Puppeteer is a web-based protocol automation system, and it's designed to alleviate the workload that synthetic biologists face when performing DNA assembly. Puppeteer frees synthetic biologists from the recurring task of planning, optimization, computation, and execution arising when they perform assembly. The Puppeteer solution performs three main functions. It first formalizes biological procedures into software. It manages resources. These are all laboratory resources. And it creates plans and instructions for the execution of DNA assembly. The inputs it requires is of 3 types. It needs a DNA assembly specification. An assembly specification specifies how to compose a target genetic construct. It also needs an assembly methods and protocols. These methods and protocols tells you and describe the procedures to follow to perform DNA assembly. And it needs resources. This is all resources that you need in the laboratory, like instruments, labware, personnel. It needs to know that information. And the output, what does it produce? It creates a plan with instructions for assembly execution. Now, we define roles in Puppeteer, and what we plan to do with this is we encapsulated different levels of expertise that are required to use Puppeteer. And it's important to note that one person may fill one or many, even all of these roles, but it's a requirement to use Puppeteer to fulfill each one of these roles. I'll go into details of these roles now that we go over this system diagram. This is a complete system diagram of all the pieces of Puppeteer. In blue, we have the roles. The roles are resource manager, developer and protocol expert, designer, instrument operator, and laboratory technician. I have a robot there as a role. I'll explain more about it later. It's not a technical role, but it performs an action of an executor. Anyway, the data is in pink, and the Puppeteer components are in purple. So I'll go first with the resource manager. It's right there. Puppeteer needs to keep track of resources. I mentioned that before. And well, someone needs to tell Puppeteer what are these resources, how many do we have, and where are they. So we do that through a graph. We call it resource instant graph. This graph is in there you can specify all the resources that are in the laboratory and where they're located. And it's the responsibility of the resource manager to introduce information to Puppeteer. Puppeteer needs also to know the assembly protocols and assembly methods that are required to perform assembly of DNA constructs. And these are introduced to Puppeteer by the developer and protocol experts. Protocol experts are, as the name says, experts on these assembly methods and protocols. They need to know these procedures to a certain level of detail, and to be capable of generalizing them, and to communicate to the developer so that they can be encapsulated in what we did, encapsulate them in protocol classes. These protocol classes are used by Puppeteer to produce plans and instructions. I'll talk about that later. We also need what is the DNA construct you wish to construct, and what are its characteristics, what is the sequence, the parts that make this construct. So this is specified in the assembly graph, and it's provided by the designer. So now Puppeteer takes the resources, it takes the protocol classes, it takes the assembly graph into a planner, and it creates 2 things. It creates instructions in form of CRIS. We define CRIS to be common human-robot instruction set. As the name says, they can be applied for robots and humans as well. And it creates a plan. It's missing an arrow here, but it creates a plan. And the plan is a set of tasks, biological procedures, with their schedule, so they have times, and they have personnel assigned to those tasks. So it tells you who's going to perform each task. The instructions are fed as input into our compiler, as well as another piece of information. It's a configuration file. Our compiler is capable of producing instructions for many types of robots. In fact, it's possible to produce instructions for any instrument that has some sort of API. And the compiler needs to know, information about these instruments. Need to know the specifics, and they're provided in the configuration file. And who provides this information? This is the instrument operator. And the instrument operator is an expert in these devices. is a person knowledgeable enough to provide this configuration class. Then the compiler produces instructions for, well, it takes execution instructions that are common for both humans and robots, but they're not ready for execution. They're not ready to be used. The compiler produces human instructions and robot instructions. And lastly, the last role is the laboratory technician. This person performs the actual assembly. They do the pipetting and all the manual labor in the lab. And they do it by following a plan that tells you what protocols to do and when to do them, and instructions. These instructions, to give you an idea, they are like cooking recipes, but instead of for food, they are for DNA assembly. In a similar way, well, the instructions of robot are scripts scripted for execution in a robot. We use the TCAN 150 provided by the customer.
So what I'd like to do now is go over a simple robot assembly example. So what you can see here is an assembly graph. This is a DNA assembly specification in graph form that just tells you I want to put parts, DNA sequences A and B together to combine AB. And what I have here is the intermediate data structure or protocol graph visualization that Puppeteer generates when it processes the assembly graph for a specific assembly method, which is the 5-brick robot assembly method. And what I have here is just the abbreviated version of the protocol graph. You see, these are robot protocols that you see in these sections, and it kind of tells you how will you process sequence A and various inputs and outputs that it follows along this trajectory until you get to your final sequence, which should be AB. Now, this protocol graph only tells you the high-level order or sequence of operations you need to perform. What actually needs to happen is that I, as the designer, will supply this information as well as some personnel. So for this example, I selected Professor Pisano and Professor Kia, and you can see that they're actually assigned to these protocols at a specific start time. And they'll take these DNA sequences, perform the respective instructions that are generated with their input, get the output, and this will translate across. And what you can imagine is that planning an assembly like this, where I'm just putting two parts together, it's kind of easy to understand and kind of compose, and perhaps you can sit down and write it out within the course of maybe a couple hours if you're new to this, or really fast if you're an expert in synthetic biology. So what I want to show you next is a slightly more complicated example. What if I wanted to do A, B, and CD? Now you kind of see how the assembly graph grows for just putting together four sequences instead of 2. And now all of a sudden this intermediate data structure has grown to a lot more steps. And this is kind of the process that happens in actual labs in synthetic biology. People actually do take the time to plan these out carefully because you don't want to waste resources, and they take the time to perform these steps accurately because it's always valuable research that's being done. And you can see Puppeteer generates this protocol graph that reflects how A, B, and CD are combined, and then maps them to a plan. And you can see how it staggers the plan so that Professor Kim and Professor Pisano can perform the 2 sets of protocols before they can do the next 2 sets, and then move on to the next set. And what I want to highlight here is that Puppeteer succeeds in alleviating the designer from planning and optimizing such an assembly. As a designer, I just had to submit this graph in text form and the 2 personnel that I want to assign to my to my project. Puppeteer also alleviates the technicians from computing resource information because Professor Pisano and Professor Kia do not have to sit down and figure out all this information and assign times and whatnot. They will just get an instruction sheet, and these times are already selected based on their schedule. So they just have to follow these fully specified recipe-like instructions for technicians with great ease. What I'd like to cover now is the Puppeteer architecture. Basically, the core of Puppeteer is written in Java. We wanted to make use of the object-orientedness of Java with the inheritance and the way we organize our assembly method classes as an interface, and the protocol classes and how we extend them and reuse them in different assembly classes. For example, you can write your own assembly method for how you would build A and B, and we can encapsulate that in our system by simply adding your Java class and letting the designer select that in their assembly graph specification. And we built Puppeteer on the WaveMaker web application framework. And we did this because for three primary reasons. Wavemaker provided simple UI implementations for a web front end. It provided easy database interaction, and we had to use our customers' MySQL database for accessing some sequence information, for figuring out which information is associated with which part, etc. And the third piece was that it allowed for easy Java integration. And this is key because the way we designed our system is that we utilized Wavemaker Java services. The three main ones that I showed here, for example, we use a database service for pulling all the data that we need for the parts, for sending data into our database. We use a file upload download service for creating files that are generated on the fly, such as that protocol graph, or for retrieving data such as the technician's instructions file. And the key service is the Puppeteer service, which houses the assembly method classes, the protocol classes, as well as the key components such as the planner that takes in all the input, generates the quiz instructions for the compiler component that needs access to all of the files, the configuration files, as well as the method that converts an assembly graph to a protocol graph. And all of this is hosted on a Tomcat server. So this is just a web archive or a WAR file that's deployed to a Tomcat server. So as a client, so as a designer, I would just I would open up my web browser, log into the client, use it. And as a technician, I would open up my web browser, log in, download my file, and then start performing my experiment.
So what were our results? Well, first of all, we managed to theoretically free synthetic biologists from planning, optimization, computation, and execution. We also managed to fulfill most of our clients' requirements. However, we did not address two of them. One of them involves the actual experiment and execution demonstration. What this means is we did not perform actual DNA assembly in the lab. And we, consequently, didn't produce experimental results within 5% accuracy because it's required to perform DNA assembly. We didn't address these two requirements because of time constraints. However, we did validate the correctness of Puppeteer. What they mean, the correctness of instructions produced, the correctness of the plan, by comparing them with the instructions and the instructions in the laboratory manual that is used in the laboratory of a client. We also performed important tests using the robot in the TICAN Evo 150. The instructions compiled and we did extensible tests in the robot, and everything seems correct. So with this, I'd like to open up the floor for any questions.
Do you have a way of sort of checkpoints, so for a topic like this, checkpoints where they are at in the sequence event? So for example, if you give me a list of 300 different steps made, at some point in 137, I forget where I'm at, can I checkpoint that easily?
Well, the way our instruction files are generated right now, they're fully specified. We do not have an interactive form of that, but I could imagine in the future revision of Puppeteer where we could have a kind of interactive instructions file where you kind of tick mark which step you performed and where exactly you were at.
How exactly do you optimize the flow?
So what we do for the optimization for planning is that we traverse the protocol graph that we construct. We perform a topological sort, and we analyze at each step level where we can parallelize the tasks. And that's why you saw that staggered effect of a more complicated graphing example.
Also to mention that all our protocol graphs are all DAX, so the topological sort is easy to do.
It can scale up with more technicians very easily, like it'll distribute the label around.
Yes, in the current implementation. And it's designed, it's a modular design or architecture. in that if you come up with a better planning algorithm, we can replace that with our current planning algorithm. And the other components will stay the same, and it'll just figure out how it will plan the remaining section. So right now it'll try to plan one personnel per protocol. But if in your lab you have personnel that can do 2 protocols at the same time, you can change the way the planning goes.
More about your custom requirements. Is this supposed to scale from single protein all the way up to a full synthetic genome, or is there kind of a limitation to what you're trying to do?
I don't know if I have enough background to answer that question completely. We were targeting specifically were BioBrick and Moklo reactions. There are limitations on the sequences that parsers can use, but I don't know that.
So you have this automated system, right? Without the automated system, presumably the designer has to do this by hand. Do you have any idea or any metrics on how much improvement your system is making, either in terms of the time or whatever?
We have brainstormed some ideas. One metric we came up with, we haven't had a chance, due to time constraints, of actually trying this out, is we could time how long it takes our system to come up with a full plan for just what I showed you today, and how long it would take actual experienced synthetic biologists to come up with that exact same plan with all the same starting information, all the same sequence information, who's available, their schedules, etc. How long it takes them to do it by hand, and probably people that are not experienced in synthetic biology, how long it would take them to kind of figure out how they would design their plan, and maybe compare those as maybe a time measure.
And then we have how many metrics?
So, kind of piggybacking, so how long does yours take?
So
How long does it take to like produce a plan?
Yeah, for you guys, for your I'm assuming they're kind of like just test sets that you're working with.
right.
Long as you can speak.
I don't know off the top of my head. The simple AB example that we always test runs in under a second, depending on internet connection, etc. The ABCD example, for example, it took a couple seconds longer because the assembly graph, actually what's not optimized is the translation from the assembly graph to the protocol graph. And the reason why this is not optimized is because you have to encapsulate the biological procedures. You have to formalize these into software. That's not always easy to optimize. So I would think that we have a bottleneck in that translation. But if, for example, I went to a really strong Java programmer and said, hey, this is what I want to do when I want to convert this graph to this graph. What's the best way to architect this? We could plug that in and improve the speed.
All the ones that we have tried, it's just been order of seconds.
I will also have missed this during your expose. How complicated a recipe can you generate?
Why do you give a recipe? Instructions.
So,
Well, it all depends on what the recipe is like.
Well, the recipe is usually in steps.
Yes.
But
So
how complicated a recipe can you generate? Do you have a limit on the number of steps or how complicated it can be?
we have no limit number of steps. We have no limit number of steps. In terms of how many instructions you produce, how many steps we have to go through, we don't have a limit I can think of right now that will limit us.
if I can jump in, like for example, when we were trying to come up with a good example for our functional test, we came up with a bogus protocol, which is taking an existing protocol, restriction digest, that just does 5 pipetting operations. And we tried to do this, I believe, 160 times. So that's just 5 pipetting operations, but 160 times. You can imagine that an instruction file or a recipe file for that would be thousands of lines long. So we do generate thousands of lines of instructions that can be performed by a robot or by human, because they're just pipetting steps. If you're talking about complicated biological procedures, I would claim that that's more limited in the bio side. So that will be dependent on how well these experts, these assembly experts and protocol experts, can generalize these such that we can encode them in software. That would be the limitation for how complex or complicated they are.
Anything else? All right, thank you.